In this microbiology lecture, we are going to explore the idea of spontaneous generation. For centuries, we didn't really understand where new life came from. So there was this idea of spontaneous generation, that life was able to come from non-living things. This early idea of the creation of life, or where life came from, was first written by Aristotle. So sometime in the 300s BC, or BCE, Aristotle theorized about this idea of spontaneous generation, that life could spontaneously arise from non-living matter. And what Aristotle said is that this could happen if that non-living material contained what he referred to as pneuma, or this vital heat, or this life force, essentially. And what he said, or kind of the evidence that he presented from this, was when fish could appear in a puddle of water, or maggots appearing on an animal. This idea of spontaneous generation was not easily let go. It continued to persist on into the 17th century. And at this time, scientists started to perform some experiments to determine whether spontaneous generation existed or this was an outdated idea. People who believed in this idea, or people who were proponents of this theory, provided all sorts of evidence. You know, one of the ones is that mice were spontaneously generated from hay that was stored in a barn. That mice could arise from rags and wheat kernels. Now, looking back on this, of course, we think it's silly. We know where the mice came from. They were attracted to the hay, or they were attracted to this wheat, and so they showed up. It was a source of food for them. But back then, the idea was, we don't really know. We don't really know where these mice are coming from. So it's possible, it's plausible that they're coming from this hay. So one of the first real scientific experiments that we see was done by Francesco Redi. And this was done in 1668, and he performed this relatively simple experiment. And what he did is he put a piece of meat in several different containers. He had 2 that were left open to the air, he had 2 that were sealed with a cork, and he had 2 that were covered with gauze, or like a cheesecloth type material. And what he found is that when the meat was left out to the environment, flies would get in, maggots would appear on the meat. In the cork-sealed container, there were no maggots. And in the gauze-covered container, there were no maggots on the meat, there were maggots on the cheesecloth. And so what he said is that maggots are not spontaneously generating. Maggots are coming from the flies. The flies are laying eggs, those eggs are hatching, and they're becoming maggots. The open container of meat drew in the flies, and so the flies were allowed to lay their eggs on the meat, maggots appeared. When the flies couldn't get into the meat, or couldn't get into the container with the meat, as in the cork-sealed container, there were no maggots. So he said, spontaneous generation doesn't exist, we're just missing those middle steps. Okay. About 100 years later, another scientist, John Needham, performed his own experiments, and he took some broth that was infused with either plant matter or animal matter. He boiled it for a little bit. His idea was that he was going to kill if there was anything living in there, that the heat would kill it. He sealed up the flasks. And what he said, or what he found, is that after a few days, the broths became cloudy. There was bacteria growing in these broths. And so he said, well, I boiled it. I would have killed everything that was living that was in this broth. So spontaneous generation must occur. Looking back on his experiment now, we realize, well, he didn't boil the broths long enough. Spallanzani did not agree with Needham. He performed hundreds of experiments of his own, basically replicating what Needham had done using the heated broth. What he found is that the heated flasks remained clear as long as they were sealed, but if the flasks were open to the air, then they became cloudy. So he said that, Needham just didn't boil his things long enough. As long as they're boiled thoroughly, then nothing's going to grow. Needham said, no, that there's this life force that exists, and when Spallanzani boiled his broth, he basically destroyed this life force. So we're still kind of going back and forth. We've got some, proponents that are really still advocating for the idea of spontaneous generation, and then we have those Reddy and Spallanzani who have really provided some concrete evidence that spontaneous generation doesn't exist. But the final nail in the coffin, so to speak, of this idea of spontaneous generation came from Louis Pasteur. And Louis Pasteur basically repeated Spallanzani's experiments, but instead of closing the flasks, he created these swan-necked flasks. So you can see here these flasks are open. They are not sealed, but there's this curve in them that essentially what happens is air can get in. Needham said, well, it's the air. Air can't get in. So air could get in, but dust and therefore bacteria would settle here at the bottom of this curve. So Pasteur said as long as the curves remain intact, so as long as this swan neck remains intact, then the broth remains clear. No bacteria are getting in there. But when the neck of the flask was broken off, and now dust and bacteria could settle directly into the broth liquid, then it would grow cloudy. So Pasteur really was the one who put this idea to rest, and not the only thing Pasteur did, but it really allowed us to completely discard this idea of spontaneous generation. Which, again, looking back on now, we think, oh, this is so silly. Of course we know that living organisms can't arise from non-living matter, but it really was something that took hundreds of years to put to rest and to really just say, okay, this idea of spontaneous generation, this is not real. Now, continuing into kind of looking at some of this early history of microbiology, we have the groundwork for modern cell theory. So the word cell was first used in 1665 by Robert Hooke. He had designed these very simple microscopes, and he was observing pieces of cork. And what he noticed is that the cork kind of had this honeycomb appearance. He said that it resembled this honeycomb or small boxes or bladders of air. And he could see these walls in between each one of these. And so he came up with the idea of a cell because they were individual little boxes, kind of like the cells in a monastery, which were the individual rooms where the monks lived. So he identified these as cells, but we didn't really understand at the time the significance of this. And so it took a long time, almost 200 years later, until we get to two individuals. We had Matthias Schleiden, who was a botanist, and Theodor Schwann, who was a physiologist. And Schleiden was looking at observations of plant tissues, and he noticed that they were described as cells. He could see these same little box-like structures that Hooke had described almost 200 years earlier. And then Schwann was basically doing the same thing, except he was looking at animal tissue. And he had this conversation with Schleiden and realized that plant tissue and animal tissue were very similar. They were both composed of cells. So this laid the groundwork for the modern cell theory, in that plants and animals were both composed of cells. Then we have Remak and Virchow, who kind of expanded upon this idea of cell theory. Remak kind of was the first to propose the idea that cells arise from pre-existing cells, that cells undergo cell division, and that's where new cells come from. Some people really questioned it. I mean, it hasn't been that long since we've discarded the idea of spontaneous generation, so people were still a little hesitant to believe that cells, could undergo division and produce new cells. 3 years after Remak published his paper, Rudolf Virchow published an essay called Cellular Pathology, which also used this idea. You see there the Latin phrase omnis cellula a cellula, which means all cells arise from cells. And his work really was very similar to Remak's. And there was kind of an idea that maybe Virchow stole some of Remak's work or plagiarized some of Remak's work. So this ion ethics box here, I'll let you read this, but it kind of talks about really who should have gotten credit for this idea that cells arise from pre-existing cells. So we have this idea that, cells are the fundamental unit of life because we see them in plant cells and animal cells. We know that animals and plants are both composed of cells, and we have this idea that all cells arise from pre-existing cells. And so that really is the framework of the cell theory. Now, along the same time that we're discovering what cells are and that they make up living organisms, we had other individuals who were starting to see the internal structures of cells, and we're starting to study the internal structures. Robert Brown observed the nucleus, which he first saw in plant cells, and then the German botanist Schimper was the first to see chloroplasts. And so then we have this idea of what is called the Endosymbiotic Theory, that chloroplasts and mitochondria used to be essentially existing outside of cells, and that cells actually took them inside and made them part of the cells. And some of the evidence for this is that mitochondria have their own DNA. This is something that hasn't really been around that long, that we've known about, that mitochondria, or we call it mDNA or mitochondrial DNA. And that's some of the evidence for the endosymbiotic theory. And then as we learned more and more about microbes, we were then able to develop this germ theory of disease, that diseases can result from microbial infection. So these organisms that we previously couldn't see and now we can see may be causing illness. And we'll stop there for this lecture.